Hello students, welcome to today's tutorial on the method of measuring microbial growth. This topic is crucial for understanding how scientists track the multiplication of microorganisms in various environments. By the end of this video, you will have a clear understanding of several methods used in laboratories to measure microbial growth effectively. Let's get started. Measuring microbial growth is essential in various fields. For example, in the food industry, it helps us ensure products are safe for consumption. In medicine, it allows us to test the effectiveness of antibiotics. And in biotechnology, it helps optimize fermentation process to maximize product yields. Now, let's dive into the methods used for this purpose. Broadly, we can categorize these methods into two groups: direct methods, where we count the cells directly, and indirect method, where we estimate growth based on changes in the environment, like turbidity or metabolic byproducts. Let's explore each category in detail. The direct microscopic count method involves counting cells directly under a microscope using hemocytometer. This slide has a grid that helps in counting cells systematically. Although this method is quick and inexpensive, it does not easily differentiate between live and dead cells unless we use special stains. In the plate count method, first we dilute the sample serially to reduce cell concentration. We then spread a known volume on an agar plate and incubate it. Each viable cell forms a visible colony. By counting these colonies, we calculate the number of colony forming units or CFUs. This method only counts live cells, making it very reliable for estimating viable populations. Another common method is turbidity measurement using a spectrophotometer. As the culture grows, the solution becomes cloudier, and the spectrophotometer measures these as optical density. The higher the OD, the more the cells are present. This method is non destructive and allows for real time monitoring, but it does not differentiate live from dead cells. The dry weight method is ideal for measuring large biomass, especially in fungi or filamentous bacteria. By drying the microbial pellet to remove water, we can accurately weigh the total cell mass. Although accurate, this method is time consuming and destroys the sample. In metabolic activity measurement, we assess the growth by analyzing metabolic byproducts. For instance, we can track the consumption of glucose or the release of CO2. This method provides real-time data on activity of the cells and is often used in fermentation process. Flow cytometry is a high-tech method that provides detailed analysis of cell populations. By staining cells with fluorescent dyes, the flow cytometer can quickly count and sort cells based on size, complexity, and even DNA content. This method is highly accurate but requires advanced equipment and expertise. Let's compare the methods based on key criteria like accuracy, time, cost, and whether they can differentiate live from dead cells. For quick estimation, turbidity is effective. For accuracy with viable cells, the plate count is ideal. For detailed analysis, flow cytometry stand out. The choice depends on the context of the experiment and available resources. In conclusion, choosing the right method depends on the type of microorganisms, the nature of the sample, and the level of accuracy required. I recommend using a combination of methods when possible to get the most reliable results. I hope this session helped clarify how we measure microbial growth in different scenarios. If you found this tutorial helpful, please don't forget to subscribe for more educational content. Keep learning and stay curious.